Okay, good afternoon everyone and welcome to today's webinar on Ensembl Release 90. I'll just boot up my presentation. So my name's Emily Perry and I'm in charge of the Ensembl Outreach team. My team does various things to promote and tell people and teach people about Ensembl. So we deliver training workshops, we do online courses and webinars and help videos. We do help emails. If you have an email Ensembl and ask for help, it's my team who'll get back to you. And we do social media and things. So in various ways, we're telling everyone all about what Ensembl is and how they can use it in their work. So during the webinar today, you should have a display that looks like either one of these two, depending on whether you're using the web interface or the little widget that comes out separately. I've muted everyone's microphones just so that we don't get kind of disturbed. There's a chat box in the interface. If you ask any questions via the interface, then there's people in the group who can get back to you. I will also check it periodically for questions. Because the chat box doesn't have any threading, just if you have asked a question, look out for people addressing you by your name in the chat box. You can also see there's a materials link in the interface. If you want to download the slides from today, you can do so there. We'll also be posting this video onto YouTube at some point over the next day or so. So what I'm going to do, I'm going to start by giving a bit of an introduction to the Ensembl, if there's anybody here who's not really familiar with it. I'll then follow it up by talking about some of the new or updated rodent genomes we've got in Ensembl, and I'll talk about two new things that we've been doing with clade-based genome annotation and also importing external annotation. I'll talk about the new pig genome. I'll talk about GENCODE, and then I'm going to talk a bit about wiggle files scaling. I've got a demo there, some updates to variation data, and some updates to our microarray data, and I'll show you a bit of a demo to see how you can use that as well. And then I'll say a little bit about some GFF file updates. So we're starting with the stuff that we think is really cool, and towards the end it's a bit more esoteric, let's say. So just as an introduction, why do we need genome browsers? Well, genomes are big, and they contain lots and lots of data, and they're being produced at quite alarming rate these days. And when we look at genomes as they are, this is basically what a genome is. It's a bunch of A's, T's, G's and C's, which in itself is a fairly meaningless thing. So genome browsers try to make the data mean something. And we're one of the major genome browsers, along with UCSC and NCBI Map Viewer, but there are smaller scale single species genome browsers out there as well. And in Ensembl we have quite a lot of data. So we have, I didn't update this slide for this new release, we have gene builds for about 100 species, not 70 species, which is quite exciting. We have gene trees, so we compare all of the genes pairwise to each other. We have what we call a regulatory build, which incorporates data from things like ENCODE, roadmap epigenomics, blueprint, in order to predict the position of things like promoters, enhancers, stuff like that. We have displays for looking at variation data, so SNPs and small insertions and deletions, as well as large-scale variation data. And we have a tool for analysing your own variation data called the VEP. We also have ways to display your own data, which we'll look at a bit when we look at the wiggle file scaling update that we've got. We've got Biomart for data export, and I'll be showing you a bit later on how we can use Biomart to find genes associated with microarray probe sets. We also have programmatic access via APIs, and we are completely open source. And you can access us at various scales, so you can look at the main browser site and the mobile site to look at genes one by one. You can look at groups of data with Biomart, the REST API, or the VEP. You can look at kind of the whole genome all at once with the FTP site, and that's where the GFF files come in that we'll be mentioning later. And the Perl API and MySQL are nice for multiple scales. Ensembl covers vertebrates, so this is a slightly out of date species tree, but we tried to cover a wide range of taxonomic space. We tried to cover species that are agriculturally important, model organisms, environmentally important as well. But as you'll see as I speak later, we're expanding our capabilities. So as we're doing this, we're being a lot less selective as to what we're putting in. Because of the way that we're expanding, instead of saying, oh, well, we have to prioritize this over this, we will find that as our capabilities increase, it would just be, is there good enough quality data? And we will be, this is going to get bigger and bigger and bigger, and I don't think I'm going to be able to fit it on a slide at some point in future. If you're eagle-eyed, you may have spotted that not everything on this slide is a vertebrate. We also have C. elegans, Drosophila melanogaster, and Saccharomyces cerevisiae, which we have in the database partly for historical reasons, but partly we use them to anchor our gene trees. By being kind of outgroups, we can anchor these trees. So we're here because we've put a release out, and this is part of our release cycle. So every 2 to 3 months, we will put out a release. So our last one was in May 2017, and that was Ensembl 89. And then over the 3 months since then, we've done various things to get us to Ensembl 90. So things that you might see in any given release cycle are new genome assemblies, which we've definitely seen in this release, underlying software updates, which we've done. We've got updated gene sets, which again we've done. Running our comparative genomics analysis on new genes and genomes. So I mentioned that we do gene trees by comparing all the genes pairwise to each other. If we've got new genes, we have to run that again. And we also do whole genome alignments. So if we've got new genomes, we need to incorporate them into some of our multiple genome alignments, but also we want to compare them to some of our well-studied species. So I'll be mentioning a lot of rodents later. They've all had whole genome alignments done against human and against mouse. Updated variation data, which we'll see there's a lot of. We don't have any updates in the regulation data this release, but we do have updated interfaces with the wiggle tracks, as you'll see. So we have new or updated rodent genomes. So here we've got 18 images of rodents. There's actually 19 new genomes, and I'll explain what that means in a moment, and 17 new species. So we have updated annotation for kangaroo, rat, guinea pig, and squirrel, and we have new genomes for a number of other species, including naked mole rat and Rickeyu mouse and Brazilian guinea pig, a number of other species. There is a full list available in the blog post, which I think most of you have probably seen. We also have 2 different kinds of annotation that we've used. So for 3 of our species, we've actually imported the annotation. So we've imported a Chinese hamster ovary, a Rickeyu mouse, and a shrew mouse. Annotation from external sources. For all the other species, we actually annotated them all in one go in what we call clade-based annotation, and I'll explain what that means in a moment. And for the first time ever, we have multiple genomes per species. So we actually have 2 naked mole rat genomes and 2 Chinese hamster ovary genomes. Note that these are Chinese hamster ovary, the cell line, rather than just Chinese hamsters. So how many genes have we got? So this is a summary of the different kinds of genes, and you'll see that there is the number of protein coding genes is fairly constant between the species. The protein coding genes are generally the most high confidence genes that we have, and they're the easiest to annotate. For this reason, we tend to get closest to the actual number as there are in a real genome in our annotation. So between these species, you can see that for most of them, the number of protein coding genes is around the 20,000 mark. The number of non-coding genes really depends on the source databases we have. And for some species, for example, the Hypocephalus, which is the naked mole rat here, you can see we've got quite a lot of link RNAs because there is an extensive amount of link RNA data out there. For other species, such as this Dipodomys audii, which I'm not quite sure what it is, there's much fewer link RNAs because there isn't so much of that data publicly available. So you will find more consistency in the protein coding genes than in the link RNAs and other non-coding pseudogenes and microRNAs and miscellaneous RNAs. So we've done our automatic gene annotation on these species. So we annotate genomes by plotting cDNA and protein sequences onto the genome using sequence similarity. So we use uniprot proteins from that species, and depending on how well studied the species is, there may be lots and lots of uniprot proteins from that species. There may be very, very few. We use cDNAs from RNA-seq. These are always species-specific. We don't use RNA-seq from a different species. And then we use Ensembl transcript models and uniprot proteins from related species. So many of these species, these rodent species we've used, were annotated using uniprot proteins and Ensembl transcript models from mouse, because this is obviously a very well-related species. So the way this works, this is a kind of summary, is we load the data in, we run repeat masking. So this is where you cover up the regions of the genome which are repeats, because this will interfere with your other data. We run something called GenScan, which just looks for open reading frames. These don't form the gene models at all, but they're just a kind of helper. And then we run 3 simultaneous pipelines. The protein pipeline, which maps on the uniprot proteins, the projection pipeline, which maps on uniprot proteins from related species. In most of these cases, this was mouse, and the RNA-seq pipeline, which maps on the gene models from RNA-seq. And then we map the data together from these 3. We combine the data, we prioritize. This one here, the protein pipeline, is considered to be the best, and this is used to produce the best possible gene set. When we're working with a clade of species and we've decided that we've got this new pipeline that does this much more quickly than we've ever done before, and we've decided the way to do it is do it clade at a time. And the reason doing it clade at a time works is because you can use the same set of homologous sequences. All of our related species sequences in this case could come from mouse. We're going to do a primate set at some point in future, and we'll use the homologous sequences from human. We're going to do a fish set, and we'll use the homologous sequences from zebrafish. So you can pick your set. You can use the same parameters to run the annotation. Because they're closely related, it means that there are similarities that make this possible. When you're doing unrelated species, you've got to set different parameters, choose a different set of homologous sequences for each one. So it's a bit. This makes it much more quicker, makes it go much more quickly, annotated by clade. We can also use a comparison between the species for our quality control. And we can do things like projection by alignment. So you can align regions and say, well, this is where we've got all the mouse models. This is where we've got, I think this is the Algerian mouse. And we can say, well, these regions perfectly align, therefore these genes probably map across. And this is an additional step that we can do to make sure that it's all working and matching up as we think it should. Another thing we've done is we've imported annotation. Again, this allows us to bring in more genomes more quickly. Now, obviously, we have quite a trusted gene set, so we don't want to be bringing in anything that is not of high quality. So we've been ensuring this quality. We have done rigorous QC on all of these data that we've imported, but the main thing is that we actually brought them in from sources that we very much trust. we brought in this Chinese hamster ovary annotation from Eagle Genomics, and it was actually done by someone who used to be on our gene build team, so we very much trust them. We also use the comparative analysis annotation toolkit from UCSC. Again, these are collaborators who we've worked with many times before, and we trust their annotation. So we don't yet have a proper pipeline for bringing things in externally, but if we have trusted sources and we know that we can properly check over it, we're quite excited to be starting to bring in annotation from elsewhere. Another thing we've done is we've now got multiple genomes per species supported within the same database structure. So we have two naked mole rat genomes, one which happens to be male and one which happens to be female, from the Broad and from BGI. And obviously naked mole rats are really exciting species to study because they have these incredibly long lifespans, they don't get cancer, and they're also proper social mammals, which is a really unusual thing. So we brought in these two genomes. We annotated both through the same pipeline, through the clade-based annotation. And the reason we did this because there were two genomes which were of suitable quality and both had been adopted by the community. And we didn't want to force members of the community who were used to working with one to have to switch over to the other because that was the one that was in Ensembl. We wanted to give people that choice. With the Chinese hamster ovary, we had a genome that we pulled in from BGI, and we had another genome that was produced by Horizon Discovery and the Wellcome Trust Sanger Institute. The one from BGI went into our clade-based annotation pipeline, but as I mentioned on the previous slide, the other one was actually imported from Eagle Genomics. So again, it's making both available to allow people working with either to stay on the same genome assembly. And we know that Chinese hamster ovary is a very popular cell line that people use in various studies. So in the future, we expect to see more genomes appearing in Ensembl. That our rate of genome release is ramping up. This is the largest release we've ever done in terms of numbers of genomes. But we expect it to be superseded very quickly because we're going to continue with this kind of pace. So we're going to do more clade-based annotation, we're going to import more genomes from trusted external sources, and where there are multiple well-used high-quality genomes for one species, we will continue to support this idea of having multiple. So another new genome that we've got is the new pig, Scrofa 11.1. This comes from the Swine Genome Sequencing Consortium, and it comes from a single Durek sow who happens to be called TJ Tabasco. This is her head, which is mounted on a wall of somebody's office who's involved in this. So this wasn't using the clade-based annotation. Pig is one of our high priority genomes because of its agricultural importance. And we did traditional Ensembl annotation using pig RNA-seq data. We used PacBio long reads, pig cDNAs, and proteins from related vertebrates, and obviously from pig as well. And we also have manually annotated genes from Havana in pig. So just to kind of explain what we mean by the manual annotation and the automatic annotation. The automatic annotation is, as I've described before, automatically plotting protein cDNAs onto the sequence using sequence matching. And this can be quite quick. This can run in a few weeks. Manual gene annotation is done by people, and it can take about half a day to do a single gene, and this uses data from databases and papers. Just to kind of compare manual versus automatic, manual annotation tends to be more comprehensive. You get more transcripts per gene, especially non-coding, and you get more genes overall, again, especially non-coding. There may be less evidence behind the manually annotated transcripts. This is because when you're doing automatic annotation, you have to kind of set a threshold. You just have to say, this is how much data we require in order to annotate this. Whereas for manually annotated transcripts, you can evaluate the evidence by quality on an individual basis, and you can say, actually, this is, even though there's not much of this data, the data is good enough. And manual annotation can be more accurate for difficult speeches such as UTR, splice sites, single exon transcripts, and things like immunoglobulins, which have got this interesting recombination in somatic recombination, which makes them very different. We also have GENCODE, which is a set that's made up of the automatic and the manually annotated genes, and it's the major gene set used by ENCODE, 1,000 Genomes, and other major projects. And this is just for human and mouse. And we have we have updates to GENCODE. Humans been updated to GENCODE 27, and we've also updated to the latest patch version. So these are the repairs on the dodgy bits of the genome. We're now on patch version 10 of GRCH38, and we're up to mouse M15 for GENCODE as well. So we have updates to their annotation. So now I'm going to switch away from talking about gene annotation and talk about wiggle track scaling. So previously, if you uploaded a custom wiggle track, it auto-scaled the y-axes. So they were always just set to whatever the maximum value was in the region that you were looking at. And this made it very hard to compare between tracks with vastly different baselines. If you look at these two tracks here, you can see that they're going to the same height, but if you look at the numbers over here, this huge peak here is at 61.67, and this highest point here is at 7, which is obviously a vastly different number. But if you were just to look at the size of the peaks, you wouldn't be able to see that. You actually have to look at the numbers. What we've done is we've made it so that you can decide what scale you want your wiggle tracks to be, and you can also change the color. So this is these same two tracks, but you can see that the difference between them is much easier to see. So I'm going to show you how this works. I'm going to jump out of my presentation for a moment. I'll just look, see if we've got any questions or things you want me to hop back and talk about. That's fine. So I've got some chipset data, and I've got a region I want to look at. So this is where the chipseq data is, and I'm going to go into human, and I'm going to hit display your data in Ensembl, and I'm going to I want this input2.bdg, so I'm just going to copy the link, put it in the box. And this is bigwig, and I'll just call this input2 because that's what's in the file name. I'm going to add the data, and it's going to think about it for a bit. While it's thinking, I'm going to copy. Oops, now I've gone into it rather than. Right, I'll copy that file name. Oh no, I've broken it. Right, well, rather than continue with something that's potentially not working, what I was going to show you was basically this slide. There is a blog post written by Ben in our team. I'll just show you the blog post because that's not showing up. If you go to ensembl.info, which does have instructions for how to do this. And there's a blog post here, adjusting custom tracks in Ensembl. You can see that there are instructions of how you can do this. So I will keep going, and I will talk about variation data updates. So we've got new variation data in from a number of sources. We've got data from UK10K, HGMD, Cosmic, NIH, TopMed, Nomad, DGBA, and DBSNP. We haven't got, so Nomad are still in the process of getting their data fully into DBSNP. So any data that they've submitted to DBSNP where they've added frequency data to existing known DBSNP variants, we now have that data. Unfortunately, anything that's new that they've discovered did not end up coming through dbSNP in time. So we've still got more data to come through from Nomad to come in. We've also got updates in mouse, dog, and cow from DGBA. We've also got updates to our phenotypes from GWAS Catalog, Cosmic, OMIM, OrphanNet, ClinVar, and Decipher, from IMPC for mouse, for RGD, for rat, and for ZFIN for zebrafish. So we have a lot more data there. And this has actually doubled the size of our variation database. So we're dreading to think how big it's going to be once we actually get the full Nomad stuff in. It's going to be huge. We also have microarray updates, so we have updates to the probe mappings for a number of species as well as to many of the mouse strains. And if you've not used our microarray mappings, I'm going to do a quick demo in Biomart to show you what you can do with a list of upregulated probe sets. So I'm just going to copy this list. So this is the names of the probe sets from this Affymetrix U133 plus 2 gene chip. I'm just going to copy this list. And I'm going to go to Biomart. And I'll do a new query. So I'm looking for genes that match up to these probe sets. So I'm going to genes, human genes, and now I'm going to filter with my probe set IDs, and it was 133 plus 2. And I'm going to get gene and transcript IDs, gene names and descriptions. So I've got gene and transcript ID. I'm going to get the gene name, and if I go into external, I can get my Affy plus 2. And when this loads, you'll see that it gives me a table. I was going to get the descriptions as well, never mind. And you can see that I can find the genes that are associated with these probe sets. So this can be very useful if you're working with a microarray and you want to see which genes are match up to your upregulated or downregulated probe sets. Biomart can be a very good way to do this. You can also go to an individual transcript and see them there. But we've updated these with our latest genes. The last thing to point out is we've altered the way we use sequence ontology terms to describe gene and transcript biotypes in our GFF3 files. This should ensure that the files are able to work with any parser that you might be using. There is a blog post on this. It's quite complicated and detailed. For most people, this is not something that you would need to know, but if you have pipelines that use these data, this is probably very important to you, and I do recommend reading this blog post. So we'll just wrap up now. If you have any questions, if you're interested in working with Ensembl, we have online courses and tutorials. These slides are in the materials section, and they'll be emailed to you after this webinar, so you can find all these URLs. We have a YouTube channel where you can find tutorials there. And if you're ever stuck on anything that you're doing in Ensembl, you can email us. We are helpdesk@ensembl.org. You can also follow us via social media, Facebook, Twitter, and I think I've mentioned the blog about 17 times by now, so you're probably aware that that exists. And you can host an Ensembl course. If you are interested in having us come to your institute and deliver a course taking you through how to use Ensembl, please send us an email. And if you visit training.ensembl.org, you'll find a detailed description of everything that's available. We also produce publications. If you do use our data, please cite us. And this is the team at our last retreat, showing off their fancy Ensembl mugs they all got at the retreat, who are all very grateful for the work they do. And this is, again, the team with their names instead of their faces, and all our funders, to whom we are incredibly grateful for making us able to produce the website and the data in the databases that we do, and also to be able to deliver free training and things like that.